Hi friends, let's go together with this IMViC test, the most commonly used test for biochemical characterization, especially of the organisms belonging to the enterobacteriaceae family. So without wasting much time, let's go with the first question, which is generally asked in a typical viva of this IMViC test. The first question, it states, state the full form of this IMViC, right? So IMViC, yes, here it goes, it stands for indole. So production of indole, you detect the ability of the organism to produce this indole. Second, it goes for M, it stands for methyl red test, which detects the production of acids, a mixed acid kind of fermentation. So M, it stands for methyl red. V, it stands for voges prauskeur test. So this particular voges prauskeur test, this particular what you call as test detects the production of acetone. And then we talk about C, which gives the citrate utilization. So this is the what you call as full form of this particular IMViC. So indole, methyl red, voges prauskeur and citrate utilization. This I, it sometimes refers to as Uncitrate. So it does not have an individual meaning as such. Second one, it states the indole is produced from which amino acid. Now you know that for checking the production of indole, you inoculate the organism in 1% peptone broth or 1% tryptone broth, which contains the amino acid tryptophan. Remember, the aromatic amino acid tryptophan. The enzyme here is your tryptophanase. So if the test organism, it has this particular enzyme, that is tryptophanase, tryptophan is degraded to produce indole, pyruvic acid and ammonia. So here the indole, it is produced from the amino acid tryptophan. So from which amino acid? So the answer here is your tryptophan. The third one, it says which reagent is used for detecting the production of this indole, or in indole test, which reagent is used and what are its contents. So here the reagent used is your COVAX reagent. So the reagent which you use for the test is your COVAX reagent. And what are its contents? Yes, it contains isoamyl alcohol. Remember, it contains isoamyl alcohol. It contains para-dimethyl aminobenzylaldehyde. Remember, so it contains para-dimethyl aminobenzylaldehyde, remember this, and concentrated HCl. So three components, para-dimethyl aminobenzylaldehyde, isoamyl alcohol and concentrated HCl make up this COVAX reagent, which is used in this particular indole test. Okay, next one, which medium is used for doing this citrate test? So it varies now from lab to lab. Some of them, they use a coarser citrate broth, where you inoculate the organism and just see whether there is turbidity or whether there is growth, whether the organism can utilize this citrate as a sole source of carbon. Some, they go for Simmons citrate agar. Here it is again, the basic principle remains the same, to detect the ability of the organism to use citrate as a sole source of carbon. But here it is the change in the color. So it goes from green to blue, then the test is supposed to be positive. So the two commonly used media for this, not both together, some use coarser citrate broth, whereas others, they go for Simmons citrate agar. Next one, the production of acetyl methyl carbinol. थोड़ा बड़ा नाम है, तो sometimes you call it as acetone, a neutral end product. So production of this acetone is detected in which test and by which reagent? So the test here is your voges prauskeur test. So this particular voges prauskeur test, it detects the presence of acetyl methyl carbinol or acetone, as I told. And what does it contains? It contains your Barrett's reagent, the Barrett's A and Barrett's B. It contains your alpha naphthol in your alcohol, and it contains a 40% solution of KOH or potassium hydroxide. So what does it contain? It contains alpha naphthol and KOH, that is potassium hydroxide. And which test? Voges prauskeur. What does it detect? acetyl methyl carbinol. What are the common results for E. coli and enterobacter aerogens? What can you expect? So generally, when you are differentiating these organisms, this IMViC test, for E. coli, it is expected that you get first two tests positive, whereas the second two tests negative. For enterobacter aerogens, it is expected that you get first two tests negative and the remaining two tests positive. Yes, so we were with this IMViC test. So here one more question which may be asked to you is what are coliforms? A very common question which may be asked in a Viva for a IMViC test, because this is the test we generally use, a biochemical test, to differentiate between the different members of Enterobacteriaceae family. So what are coliforms? The running technical definition goes that these are gram-negative organisms which are rods, which are non-spore formers. So those which do not form the spores, which may be motile or non-motile. So organisms in this particular category include both motile as well as non-motile organisms. Those which can ferment lactose. Yes, this is the primary property. So they ferment lactose for production of both acid and gas. So there is production of both acid as well as gas at 35 to 37 degrees Celsius. So this is the running definition of coliforms. Gram-negative organism, rods, non-spore formers, motile or non-motile, fermenting lactose at 35 to 37 degrees Celsius for production of acid and gas. One more confusing term here it may be introduced, that is fecal or coliforms like E. coli. You differentiate them from other common coliforms. Like here they have asked for certain examples. So you have organisms like citrobacter, you have organisms like Klebsiella, enterobacter, E. coli for sure. These come under the examples of your coliforms. So fecal coliforms differentiated by your elevated temperature test, where you test the ability of this organism to ferment lactose at higher temperatures at around 44 or something like that. Growth of these organisms on some special culture media, their cultural characteristics like having a metallic sheen on the EMB agar. These may be some of the tests which may differentiate between fecal and non-fecal coliforms. We will discuss these in detail in next videos. Second question goes, why does Simmons citrate agar, it turns blue for a positive test? So citrate test basically for seeing whether the organism can utilize citrate as the sole source of carbon. So how the organism will go about it? It will have this citrate. We'll check whether the enzyme citratase is present in the organism, which will degrade the citrate to oxaloacetate and pyruvate. Oxaloacetate and acetate, sorry. This particular oxaloacetate, right, will be then what you call as converted to your pyruvate and carbon dioxide. So whether the organism has the ability to degrade or use citrate as a sole source of carbon. So citrate degraded to OA and acetate, OA will be further oxidized to form pyruvate and carbon dioxide. If it is a coarser citrate medium, you will see there is turbidity. If it is a Simmons citrate agar, as it is mentioned here, so there you will see that whether there is this alkaline product which is being formed. So this alkaline product will convert the bromothiamol blue, which is the pH indicator in the Simmons citrate agar, which will turn from green to blue for a positive test. So formation of these alkaline products, they will indicate a positive what you call a citrate utilization test due to the indicator bromothiamol blue, BTB, right, which gives a bluish color in a alkaline pH range. Next, the test says what is detected in methyl red test. So in methyl red test, you are detecting the mixed acid fermentation. So when you inoculate the organism in a GPB broth or your MRBP broth, what you call. Now in these particular broth, when you inoculate the organism, whether this glucose is being fermented to form different kinds of acid, so that the pH, it gets lowered below 4.4 or 4.2, something like that. pH, if it goes below this value, then after addition of methyl red, you will have a red coloration. So you detect the formation of different acids in methyl red test. Okay. How is the pink color formation seen in a positive voges prauskeur test, VP test, what you call. So in a voges prauskeur test, you are detecting the formation of acetone. So if the organism is positive for the same, glucose will be converted to your acetone. This particular acetone in turn, in the presence of the Barrett's reagents which you add in the presence of KOH and oxygen, this will be what you call as converted to diacetyl. And this particular diacetyl in the presence of guanidine ring. So guanidine component of the peptone. So the guanidine component of the peptone, it will interact with what you call as the diacetyl formed, and in the presence of alpha naphthol, a part of the Barrett's reagent, there will be formation of this pink color. So this pink color formation is because of this acetone. So glucose, acetone, diacetyl, guanidine component interaction presence of alpha naphthol to give a pink coloration. This will indicate a positive VP test. The 2,3-butadiol fermentation pathway, whether the organism is utilizing or not. So if there is this 2,3-butadiol pathway, there will be formation of this acetone and a pink color formation indicating a positive VP test. Okay, so what are the indicators of a positive IMViC test? Okay, so if you are talking about a particular IMViC test, you have 4 test tubes, right? All inoculated with your different broths, different media. The last one, it may be a solid or a liquid medium, right? So in a typical first test, that is your Indole test, you add xylene, right? So here in this particular Indole test, after incubation, inoculation, we check whether there is what you call as formation of a pink ring on this particular medium after incubation. So after inoculated and incubated for 24 hours, and after addition of xylene, if there is formation of pink ring after addition of Kovacs reagent, then this test is supposed to be positive. So Kovacs reagent along with this gives a pinkish coloration if there is presence of indole. The next test which you do is your MR test, as we saw earlier, that here if you have inoculated the organism, if the organism is undergoing mixed acid fermentation, after addition of methyl red, there will be formation of a pink coloration due to acidic pH going below 4.4 to 4.2. Here the next one which we call as VP test, detecting the presence of what you call as neutral products like acetone. If you mix it with what you call as your Barrett's reagent, Barrett's A, Barrett's B, KOH, alpha naphthol. So here also you will find that there is a pinkish coloration. So pinkish coloration will indicate a positive VP test. For citrate utilization, if you are using a coarser citrate broth, then if there is a turbidity after incubation, if there is a growth having citrate as a sole source of carbon, then this may indicate a positive test. For a Simmons citrate agar, which is green at the beginning, formation of blue coloration, right? Due to alkaline products, it indicates a positive test. So indole, formation of pink ring after coax reagent, MR red after methyl addition, voges prauskeur pink after Barrett's reagents addition, and coarser citrate में turbidity, and in Simmons citrate blue coloration, they indicate a positive test. So stay tuned with me, Professor Girish Kukreja, for more such videos and interesting things in microbiology. Thank you.